Okay, we'll go ahead and get started. Good morning, everyone, and thank you so much for joining us today for the Master's Defense of Erika Alvarado. Erika finished up her BS in Ecology and Conservation Biology from the University of Idaho in the spring of 2019. And she was a research assistant studying harmful algal blooms during that time, and so became interested in phycology during her undergraduate work. Prior to that, she was a Doris Duke Conservation Scholar for 2 summers and also an REU working at Bigelow Labs. And then after she finished up her BS degree, she worked for Oregon Sea Grant as a summer intern before starting a graduate program here in the fall of 2019. So she joined the Marine Biology graduate program as a master's student at that time. She's also a member of the EECB program. And during her couple of years here, she has worked both as a research assistant studying the mesophotic algal systematics of the Hawaiian Islands and also as a teaching assistant for undergraduate biology labs. And so Erika is here today to tell you more about the work that she's been doing for the last couple of semesters in the lab. And so at this point, I will turn it over to Erika for her presentation.
Okay, hi everybody. Let me share my screen here. Okay. Is everybody able to see that? Okay. Okay, great. So hello, my name is Erika Alvarado. I'm presenting my thesis presentation today, which is titled Uncovering Diversity in the Mesophotic Zone of Hawaii, Species New to Science in the genera Halopeltis and Leptofauchea. Both of these genera are in the Rhodymeniales order, which is in the larger phylum Rhodophyta, which is also known as the red algal phylum. Okay, so before I get too deep into it, I want to give you a general outline so you'll know how this presentation will be structured. It's going to follow a pretty general format similar to most published papers. So starting with the introduction, I'm going to give you some background on mesophotic coral ecosystems, on the taxonomy for the taxa that I'm going to be discussing, and also background on some biogeography. Then I'm going to go into the methods, the results, and then end of the discussion about my results. And at the very end of the presentation, I will take your questions. So mesophotic coral ecosystems, they're also known as MCEs. So if you hear me saying the acronym MCE, just know that I'm referring to these mesophotic coral ecosystems. They generally range from 30 meters in depth to up to 150 meters in depth. And they're characterized by having this light limitation. So since they are deeper in the water, the light doesn't penetrate as far. And the deeper you go, the less light there is. So this zone has also been called the twilight zone. And it's pretty difficult to access because it's so deep in the ocean. People use remotely operated vehicles to get to the lower depths. They use submersibles, and also use advanced scuba diving techniques such as rebreathing technology. Part of the reason, because it's so hard to get to these depths, means that there aren't very many studies in this zone since it's hard to access these depths. There's a general lack of studies for MCEs. So the lower limit of mesophotic coral ecosystems coincides with the 1% irradiance limit. So at the surface, you have a certain percent of irradiance. And as you go down in depth, once you hit that 1% of the surface irradiance, that's actually the lower depth range for the mesophotic coral ecosystem. And irradiance is just a measure of light, if you didn't know that already. But because the depth range is based upon this 1% irradiance mark, depending on where you are in the world, these depth ranges will change. So for example, in tropical areas, the water's clearer and the light can penetrate deeper. So the depth range will be generally greater than that of more temperate areas. Despite there not being much research in MCEs, most of the reef habitat in the world is actually found in these deep systems. So 80% of coral habitat is actually found in these mesophotic coral ecosystems. So our general understanding of how coral ecosystems work is very biased towards the shallow water systems that have a lot more studies done on them. Within these mesophotic systems, the organisms there are adapted to these low light conditions. So if you see this photo on the right is actually an image from the Hawaii mesophotic zone. And you can see that the light is not too bright. It's kind of murky down there, but despite this, there are still photosynthetic organisms present, such as these algae here. These systems are pretty unique, mostly because what we are learning about them is new to us, since they are not studied. While there's a general lack of studies overall for these systems, with most studies focusing on coral and fish, there's an even greater lack of studies for organisms such as macroalgae and invertebrates. So mesophotic coral ecosystems tend to have high rates of endemism, and that's also seen here in Hawaii, where we are finding tons of undescribed species within Hawaii's mesophotic coral ecosystems. Seaweed is very abundant in Hawaii's MCEs and has been found as deep as 160 meters. And it has been hypothesized that since we are in a tropical climate, as you go deeper, the water gets cooler. So these mesophotic coral ecosystems might have the ability to house organisms that are tend to be found in more temperate areas, even though Hawaii is a tropical climate. And despite differences of shallow and deep water habitat, there are a few species that are shared between the shallow and deep coral ecosystems. And this brings me to the DRRH, which is the deep reef refuge hypothesis. This hypothesis centers around the idea that these deep reefs can be a refuge for shallow water species when they are stressed by anthropogenic disturbances. The tenets of this hypothesis is assuming that these deep reefs are more protected from anthropogenic disturbances compared to shallow reefs, and that these deep reefs can serve as a reproductive source for shallow reefs after disturbance. So the second tenet, that they can serve as a reproductive source, assumes that there are shared species between the deep and the shallow water coral reefs. And this hypothesis hasn't been fully debunked yet, but there is some mixed results. There tends to be different species in the mesophotic and the shallow water, but there are some species that are shared between both of them. I thought it was important just to mention this hypothesis due to the fact that I am studying organisms in the mesophotic zone, and just because this hypothesis hasn't been fully debunked yet, it's important to mention. So before I get on to the taxonomy for the groups that I'm looking at today, I want to give you some general background and some general vocabulary words that will really help you understand what I'm talking about. So I've numbered this figure here 1 through 5. So starting at number 1, algae has a pretty complex life history. So this number 1, this gametophyte stage, is the stage of algae that actually houses the reproductive characters. So it's haploid, and so these gametophytes either have male reproductive characters, such as the spermatangia, or female, such as the carpagonium. And it's important to remember these roots, so sperm, male, carp, female. Just remember those root words as I go along through the presentation. So when the spermatia reaches the female part of the female reproductive system, they come together and create a diploid zygote, which is the carposporophyte. And then this carposporophyte, which is diploid, will create these diploid spores, which they'll go on to create the tetrasporophyte. So when you're in the water swimming around and you see algae, what you see is usually either the tetrasporophyte form or the gametophyte form. And sometimes they look exactly the same, sometimes they look different from one another. It really depends on the species, but it's important to keep in mind that the tetrasporophyte is different than the gametophyte, and they have different ploidy as well. So tetrasporophyte is diploid, while the gametophyte is haploid. The tetrasporophyte will then produce these tetrasporangium that create tetraspores, which go on to complete the life cycle. So these tetraspores will result in the gametophyte stages. Similarly, here's a little bit more background on some vocabulary words I'm going to be using. These images to the right are actually cross sections of algae. Image number 12 is just a close-up of a blade. So I'm going to be talking about these medullary cells, and you actually see these cells in box number 11. So they're the large, round, clear cells in the center of this image. Cortical cells are the smaller, darkly pigmented cells on either side of the medullary cells. So this image in number 11 is actually a cross section of an algae blade. So the cortical cells are found on the top and the bottom of the blade, and the medullary cells are found in the center of the blade. In image number 12, you can actually see these sori spots. So this is the area that houses the paraphyses, which you can see in image 13, 14, and 15. These paraphyses are long finger-like extensions, which arise out of the surface of the blade. And these paraphyses house the tetrasporangia. So you can tell this is a tetrasporophyte specimen because it has paraphyses and it has these tetrasporangia. And it's easiest to see the tetrasporangia in image number 13. It's that dark oval shape within the paraphyses. So now that you have a general background on red algal vocab words, I'm going to get into the taxonomy. So both of the genera I'm looking at are within this larger group known as the Rhodymeniales, which is an order of red algae that's very large and has 400 plus species within it. This order is distinguished from other orders of red algae based on distinct procarpy. So if you remember that root word carp relating to the female reproductive structure, procarpy just means that the carpogonial branch is situated next to one or more auxiliary cells. An auxiliary cell is just the cell that receives the zygote nucleus. So it is the place that receives the nucleus after the spermatia reaches the female reproductive structure. And within this order, there are 6 different families. The 2 that I'm interested in are the Fauchaceae and the Rhodymeniaceae. Distinctions between the different families is mostly based upon structures that are present in one and not the other, and also based a lot upon reproductive characteristics. So the Rhodymeniaceae is the largest family within this order, and it has 193 species. If you look to the right, this is an example of what a Rhodymeniaceae from Hawaii looks like. This is one of the species I'm working on. It's the genus Halopeltis. Halopeltis has a really complicated taxonomic history. It used to be taxonomically accepted, and then that was redacted. But then recently it was resurrected or brought back to life as of 2010. So it is now currently taxonomically accepted. Once again, it has 9 different species that compose this genus. So it's actually fairly small. The characteristics of this genus is the presence of intercalacating cells, which are small cells found in between the larger medullary cells. There are typically 2 rows of medullary cells. The tetrasporangial sori, so those patches of sori, are produced either on one surface of the blade or they can be produced on both surfaces, so the top and the bottom. And they're also characterized by the presence of protuberant cystocarps. For the Fauchaceae, that's just the 3rd largest family within the Rhodymeniales order. And Leptofauchea is the genus I'm interested in for this family. These images on the top, middle, and the lower right are actually representatives for Leptofauchea from the Hawaii mesophotic zone. Currently, there are 9 species in Leptofauchea, with the newest being added last summer of 2020. So the most recent addition was described from Western Australia and is known as Leptofauchea lucidea. Characteristics for this genus include dichotomous branching, 2 to 3 layers in the medulla, and the presence of small cortical cells, and also the presence of this structure called telearachnoidea. And when you think of the word arachnoidea, you probably think it sounds a lot like arachnid, and that's for a reason. So this structure actually looks like a net spiderweb-like structure. It's a tissue that's found there, the female reproductive structure. Before I get into the general biogeography, I want to give you an idea of what the ancient Hawaii ocean was like. Probably because I am doing a taxonomy study, I'm interested in long scale evolutionary patterns in general. So about 40 to 15 million years ago, Hawaii's ocean was actually more temperate in climate. So the temperature of the ocean ranged from 16 to 20 degrees Celsius. At this time, 40 million years ago, there were some islands present in the very northwest portion of Papahānaumokuākea, but over this long time span of 40 to 15 million years ago, more islands started popping up within this time frame. Now, this temperate ocean is a lot different from what we see today. Today, it's a tropical climate, and ocean temperatures are typically higher than 25 degrees Celsius. But macroalgae is one of the most ancient marine organisms that exist. So during this time period, macroalgae may have been living within the shallow reefs in Hawaii when the climate was temperate. So when the temperature started changing to the more tropical climate that we see today, these deep ecosystems and these deep reefs may have provided refuge for these algae that used to be in the shallow water that are used to more temperate conditions. And this idea is sort of similar to the glacial refugia hypothesis, except that the glacial refugia hypothesis operates on smaller timescales, so thousands of years. And it is different from the deep reef refuge hypothesis because that hypothesis has to do with anthropogenic disturbances. The Hawaiian archipelago is the most isolated archipelago in the world, and due to this, it has high degrees of endemism. There are species found here that are found nowhere else in the world. Combining that high rate of endemism in Hawaii in general with the high rate of endemism found in MCEs, it is no surprise that endemism in Hawaii's MCEs are also high. So endemism for Papahānaumokuākea for fish in MCEs in Hawaii have been found to be 100%. So that means every fish at Carey Atoll in the mesophotic system that was encountered was unique to Hawaii and found nowhere else in the world. Getting back to algae, algae is distributed based upon different temperature ranges, and it's able to move based on physical movement of the plants and of the spores. To give you an idea of where the genus Halopeltis that I'm interested in is distributed around the world, you can see on this map that most of them have been reported from Australia, and that's because most of the studies are done in Australia for this genus. It has also been reported from South Korea, Japan, and from Bermuda and South Carolina, with our species in Hawaii being present as well. And all of these dots, I just want to know, are verified, so they have molecular representation as well, and not just, they aren't just morphological descriptions. So for Leptofauchea has a pretty wide range. It's found on both the east and the west sides of the Pacific Ocean. It's also reported from Australia, reported from the Indian Ocean on these islands called the Cocos Islands, reported from Spain and Chile, from the Gulf of Mexico. So this biogeography question brings up this broader question: how could algal ancestors arrive to Hawaii? It's unlikely that these spores are being transported long distances from one landmass to another due to their short viability and also their tendency to sink. It's more likely that these algae are hitchhiking or floating to Hawaii by hitchhiking on other marine organisms or by attaching to substrates such as wood or floating plastic that would then arrive here via currents. It is also possible that algae was transported here directly through either intentional introductions or unintentional introductions. So they can be introduced unintentionally through ballast water from shipping or through the aquarium trade, or they can be introduced intentionally, like this gorilla ogo on the left that was introduced for the agar industry. So if these algae are able to be transported via current by hitchhiking, they have to travel along these general currents. So this image here is actually an image of the North Pacific Gyre, and you can see it encapsulates the Pacific Ocean and it runs clockwise. If you can see my mouse here, Hawaii is actually positioned kind of in this area here. So it's not in direct path of the main currents. But despite that, we still see a lot of associations with algal taxa in Hawaii, with algal taxa in Asia and Australia. So this means that there are probably smaller, more local currents having an effect if these algae are more related to or able to arrive to Hawaii and not come from a main current. In this image, you can see the SCC is the subtropical countercurrent and the HLCC is the Hawaiian lee countercurrent. Both of these currents run west to east, and the North Hawaiian Ridge current runs north along the Hawaiian Islands. So to the methodology of my experiment, all of my samples were collected from the Hawaiian mesophotic zone. So they were collected from these deep reefs. From there, they were preserved as either dried in silica vouchers or pressed as herbarium samples. And from these preserved specimens, I went 2 different rounds for methodology. So I did a morphological analysis and a molecular analysis. From the preserved samples, I extracted the DNA, conducted PCR, did some sequencing, and then analyzed my results. And on the morphology side, I did some general observations of the gross morphology, did some cross sections, staining of aniline blue, and compared the morphology of the Hawaiian specimens to species that are already currently taxonomically accepted. By using both these methodologies, I came to the final conclusion of what these samples were that were collected from the Hawaiian mesophotic zone. And by using molecular analysis, it makes up for potential cryptic diversity that might occur if morphology was used alone. And by using morphology, it makes up for the potential of certain species not being represented in molecular databases. So by combining both of these methods, it's actually a very successful way to come to a conclusion on what specimen you're looking at, where it belongs, what species is it. So this is a map of my samples and where they were collected from. Leptofauchea is this bright lime green dot and Halopeltis is this bright hot pink dot. Leptofauchea has 1 sample collected from the northwestern Hawaiian Islands, with all other Leptofauchea samples being collected from Penguin Bank, which is off the west coast of Molokaʻi. Halopeltis was collected from the Big Island from 2 places in Maui and also off of Penguin Bank. Sample collections were done with the assistance of the Hawaii Undersea Research Laboratory and the Pisces submersible. So the submersible actually went down into mesophotic depths and collected algal samples in Papahānaumokuākea. Rebreathing scuba technology is what was used to collect that Leptofauchea sample. For the molecular methods, I use PCR, which is also known as polymerase chain reaction. This amplifies one section of DNA that you're interested in and it creates multiple copies of it. So you're able to send it for sequencing and find out what that section of DNA is made of, what nucleotides are present. The first step for figuring out what specimens I had, I used this thing called DNA barcoding, using the cytochrome oxidase subunit 1, also known as the CO1 section of DNA, which is extremely useful for differentiating between different species. So it's useful for algae, it's useful for insects and animals, and it helps you know what you're working with. So it gives you a good starting point to determine what other markers would be useful for you to work with. If you look on this image on the lower right, you can see each line of nucleotides is actually a separate Leptofauchea species. And these nucleotides are representative of the CO1 marker. So this is a portion of that marker for different Leptofauchea species. And you can see that there are some differences between the species, but the differences are not too great that they're uncomparable. On the other hand, these sequences do not match each other exactly. So if you found a portion of DNA that was completely conserved, it would be really difficult to find differences between species. But The CO1 marker kind of has hit the not too hot, not too cold area of the DNA. We're actually able to determine species differences within a genus. So the other markers that I use for my samples is this LSU marker, which is also known as the 28S ribosomal DNA, and the ribulose 1,5-biphosphate carboxylase large subunit, which we usually just refer to as the rbcL marker. Once successful PCR was amplified, I sent the sequences off to the facilities here on campus, the Advanced Studies in Genomics, Proteomics, and Bioinformatics, ASGPB facility, or I sent it to GeneWiz. I did analysis for my data through Genius Prime and also Cypress. So Genius Prime is a genetic program. These images to the right are actually from Genius, and Cypress is an online tool used for conversions and statistical tests on molecular alignments. Through Cypress, I ran a J-model test too to figure out what model was best suited for my data. And I ran both Bayesian and random accelerated maximum likelihood analyses on my alignments. For the morphological aspect of my project, I looked at the morphological characters through this very impressive Zeiss axioimager microscope that has a camera mounted on the top of it. I became very well acquainted with this microscope over my time here, and I used it to take pictures and look at cross sections of my algal specimens, which were stained with aniline blue to accentuate the different features within the cross sectioning. This chart on the bottom left shows you all the different morphological characters that I explored through this cross sectioning and staining. So now to the good stuff. So this is the phylogenetic tree for Halopeltis, the whole genus. On this tree was created using both CO1 data, so the barcoding marker data, LSU sequences, and rbcL sequences. All species of currently taxonomically accepted species within Halopeltis are represented on this tree, as well as a couple undescribed species, which you can see are represented by this SP1WA, SP1SA, SP2, SP2. So all these things that do not have specific epithets attached are undescribed species of Halopeltis that we have data for. Halicursus was used in the outgroup for this phylogeny. You can see on the top, these are the Halicursus. The Hawaiian samples are circled in this purple rectangular box. So these ARS numbers are all the Hawaiian samples that Halopeltis found. If you look at the nodes on each branch, you can actually see the support values. So the first value is the maximum likelihood support value, and the second value is the Bayesian support value. A maximum likelihood value of 100% indicates full support, and a Bayesian value of 1 indicates full support. An adequate threshold for support is 70. So that's the statistical significance that we use to say, okay, this clade is supported enough. And you can see at the node here of the Hawaii species, the maximum likelihood value is 81, and then the Bayesian support is 1. So this clade here is supported adequately by the statistical tests. Now going on to describe this species, I looked at it under a microscope, and I also looked at its gross morphology. So these plants range from 10.5 centimeters high to 7.3 centimeters wide. While they look like a bunch of individual branches that were just pressed on top of each other, some of these branches are actually connected through cell connections. So they're anastomosed to each other. They are actually connected. In situ, these specimens look tangled in appearance. They're subdichotomously branched, and they have constrictions present under the branching points. As you can see here, there are constrictions present in the specimen. Image number 20 is actually the surface of the blade. So I just took the blade, put it on a slide and looked at it under the microscope with the aniline blue stain. The dark small circles you see on the top are the cortical cells, which are easily visible on the surface of the blade. Looking at image 21, 22, and 23, you can see the medullary cells, which are the large clear cells in the center of the blade. And the cells are axially elongated, or they're rounded, irregular in shape, and they go up to 117 micrometers long to 79 micrometers wide. The medullary cells are present in 2 to 3 layers, while the cortical cells surrounding the outer edges of the blade are present in 1 to 2 layers in the outer cortex with an incomplete inner cortical layer. If you see image 22 and 23, you'll see this mucousy light blue substance on top of the blade, and that's actually indicative of a tetrasporophyte specimen. But these specimens were too young to have produced paraphyses, which are those long finger-like extensions that come out of the surface of the blade, or tetraspores. So while this is a tetrasporophyte specimen, I was unable to get images of the paraphyses and tetraspores. Now for Leptofauchea, there are 4 clades that I'm going to refer to based on the color of box that's around them, just for ease of discussing them. So I have a clade here surrounded in red, blue, yellow, and green. This tree is similar to the Halopeltis tree, was created using CO1 marker data, LSU data, and rbcL data. The outgroup for this Leptofauchea tree is Weber van Fassia, which you can find at the top of the phylogeny. And the support value and adequate support value for Leptofauchea is also 70 for maximum likelihood and 0.7 for the Bayesian analysis. So you can see here that we only have 1 specimen for this red clade for this ARS 09897 and 1 specimen for this yellow clade down here. For this reason, these 2 species will not be described until more vegetative material is acquired from future cruises. Looking at this blue clade actually has full support. And this clade is separate from all other species which are represented on this tree, meaning that this clade actually represents a species that is new to science. This bottom clade, this green clade here, actually encapsulates Hawaii samples, but in the middle of this clade, there's this sample Leptofauchea species 1 WA, which is from Western Australia. This is the most recently described species, Leptofauchea lucidea. So that means that all of these species gathered from Hawaii are actually this Leptofauchea lucidea species. So when describing the blue clade from Leptofauchea, I looked at the gross morphology and found that these plants were pretty large. They were 21 centimeters, or up to 21 centimeters in length and up to 18 centimeters wide. They lacked a distinct stipe and they had loosely dichotomous branching. This genus and this species also has constrictions present as well. As you can see below branching points, there are these constrictions. Something very characteristic of this species is the presence of these long internodes between branching points, which makes them look very lanky and kind of spread out. Going to the cross sections, if you look at image number 7, you can see the medullary cells, which are present in 2 to 3 layers. They are irregularly arranged and they grade into an incomplete inner cortical layer. Looking at image number 5, image number 5, that's actually the surface of the blade, and you can see these small darkly stained cortical cells on the surface. By adjusting the fine-tuning knob, I was actually able to see the medullary cells underneath. So if you look at image number 6, that is the result from adjusting the fine-tuning. I was actually able to see the clear, large cells in the center of the blade from the surface view. Image number 10 will show you the sori patch that was present on top of the blade for one of these specimens. So this patch is actually where all the paraphyses and the tetrasporangia are housed. So if you look at image number 8 and 11, you can see the paraphyses coming out of the surface of the blade, and they are 4 to 7 celled paraphyses. The tetrasporangia you can slightly see in image number 8, these dark big circles, but you can see them better in image number 9. So These tetrasporangia are tetrahedrally divided and pretty large in appearance. Naming for these species, so I would like to have the Halopeltis species and the Leptofauchea blue clade species named, and we're collaborating with the NOAA Papahānaumokuākea Cultural Working Group to acquire appropriate specific epithets for these species, which are under discussion. They take into account things like what the algae looks like, where it was found, what relationships it has. This method was used for past species in my lab, such as my lab mate's Risa's most recently published paper for this species on the right here, Rhodymeniales. So the specific epithet was actually created by the cultural working group, and it gives a land-based and Hawaiian culture significant connection to these species which are found here. For Halopeltis, all Hawaiian Halopeltis collections are distinct. They end up in one clade, so they're a single species. Halopeltis is actually a new genus record for the Hawaiian Islands, so it has never been found before in the mesophotic zone. It hasn't been reported from the shallow water. These Halopeltis collections from Hawaii are also the deepest collections reported for this genus overall, and with the deepest collection being 104 meters. There are varying morphologies from these Hawaiian Halopeltis collections, which is likely due to the location and time they were collected, since I have samples that were collected in 2015, 2006, and 2009. If you see these two images on the top of the right here, you can see they actually look quite different from one another, but they are the same species. Similarly, these images on the bottom, 39, 40, and 41, these are actually also the same species. These are Halopeltis verrucosa from Saunders McDonald 2010. So while they are the same species, having gross morphology and cryptic morphology within a single species is not unheard of. The Halopeltis species from Hawaii is considered endemic to Hawaii until we find out it possibly might be observed from other localities. It's considered endemic here. So for Leptofauchea, some of the Leptofauchea mesophotic collections are distinct and species new to science. We do have Leptofauchea as a new genus record for Hawaii as well. So Leptofauchea has never been before reported from the mesophotic zone or from the shallow reefs in Hawaii. There's one new species record of Leptofauchea. So the green clade, which encapsulated Leptofauchea lucidea, is also found here in Hawaii. The deepest collections for this genus are from our collections in Hawaii, with the deepest being 84 meters. There are other species of Leptofauchea that are found at mesophotic depths, which is super interesting that it's not only here, it's found in mesophotic depths. The deepest depth, though, for these collections is 75 meters for Leptofauchea corlingia. So that's the deepest collection for other Leptofauchea species. For Halopeltis, it was collected over a relatively small range in Hawaii. So it was collected on Big Island, and the most western point it was collected was at Penguin Bank. It's sister to the species Halopeltis adnata, which is present in South Korea and Japan. And that begs the question, how did it arrive here? So it's possible that it hitchhiked, that it hitchhiked here from South Korea or Japan and arrived here via the North Pacific gyre or smaller local currents. And it might also be the result of a past introduction that has since evolved into its own separate unique species. This is yet another link to Asian macroalgal flora from the Hawaiian Islands. So biogeography for Leptofauchea is pretty interesting. We're finding this Leptofauchea lucidea in Hawaii when it was described off the west coast of Australia. In Australia, it was described from the lower to the shallow, from the shallow reef to the lower mesophotic zone, so 5 to 37.1 meters in depth. But in Hawaii, it's only known from the mesophotic zone, and the deepest collection of this was 84 meters. This Leptofauchea lucidea that's present in Hawaii was collected in Penguin Bank and also in the northwest portion of Papahānaumokuākea. The Leptofauchea blue clade, which is described herein, is only known from Penguin Bank. Its closest relative is from the Indian Ocean, and its closest relative is called Leptofauchea cocosana, which is from the Cocos Islands that you can see outlined in this image here. Both of these taxa are not closely related. And similarly, the different clades collected from Hawaii aren't most closely related to one another, meaning that there wasn't some ancestor for Leptofauchea that arrived in Hawaii and has then since speciated to these multiple species. It's actually possible instead that there were multiple colonizations of Leptofauchea to Hawaii that have arrived and then since diverged into their own unique species. Looking at biogeography for these two genera has highlighted a general lack of algal distribution data, and specifically for Hawaiian biogeography, a lack of data for other Pacific islands as well. Having a better understanding of algal biogeography would warrant a better understanding of the presence of different algal species in this large expanse of the ocean in between Australia and Hawaii and Asia and Hawaii. This will help us find missing links between those two landmasses in the Hawaiian Islands. But in the meantime, focusing on Johnston Atoll, which is a gateway for species to arrive to the Hawaiian Islands, may be a good start. So the significance of this study is that to understand a ecosystem such as a mesophotic coral ecosystem, which we don't have much knowledge of, the first step to understanding this is to first know what is present in these systems. So just to know what species are even present there in the first place. Figuring out what species are present in these zones will help us fill in taxonomic links that will help answer questions about biogeography. Also, algae within itself is significant because it is a large component of the food chain. It feeds organisms such as urchins and crabs and fish. And studying algae within this zone also begs some more questions about physiology. So how are these algae able to deal with these low light levels when they are photosynthetic individuals? And how does that affect them in other ways? Also, understanding what sort of algae species are present is extremely important. And I can highlight this from a recent study that was done in the northwestern Hawaiian Islands, where there was this macroalgae that has overgrown multiple corals and smothered corals and fish and has basically decimated whole ecosystems up in this relatively otherwise pristine and untouched area, since it's a large protected area. That's usually what we think of it as, but there's this algae that came in here and this has caused this large scale damage. And the problem with this is we don't even know what species this was when we came across it the first time. So it's very hard to combat an algae that you know nothing about, not even its name. So it's also very important just to have an idea of what these algae species are, so that if something like this ever happens in the future, we'll have a better idea where it came from, maybe possible ways to manage the species. But for now, we're not even sure if the species is invasive or not, because we're not sure where it came from, because there's no record of it. Furthermore, mesophotic coral ecosystems should be protected because they house all this undocumented diversity. And they should be researched and protected before negative effects from climate change and acidification causes effects down in these deep reefs as well. So finally, I would like to acknowledge my advisor, Allison Sherwood, and everybody in the Sherwood Lab, my committee members, Celia Smith, Cynthia Hunter, NSF for funding this project, the Marine Biology Graduate Program and the Marine Biology Graduate Program cohort, the Papahānaumokuākea Cultural Naming Group, and also my friends and family. And thank you. I will take your question.